This video goes through how the heart action is initiated and coordinated, focusing on the roles of the SAN and AVN purkyne tissue and the myogenic nature of cardiac muscle. Won't be going into any detail about hormone or nervous control at this point because this topic does come up again later on. Okay, so starting with a similar diagram to one that you will have seen before, the only difference here being that some of the additional structures involved in coordination of a heartbeat are shown on here. I'm just going to add a label on here just to show that this bottom point of the heart we refer to as the apex. You could, if you wanted, add additional labels on for the rest of the heart, but I think it would probably get quite cluttered if you did that. Our first label here then is the sino-atrial node, or the SAN. Now this is referred to as the pacemaker of the heart because it's the SAN really that sends out waves of electrical impulses regularly about every 0.8 seconds, but that can be sped up or slowed down by the nervous system. The wave of electrical impulse that is released by the sino-atrial node will spread out throughout the atria, and that will cause the atria to contract. The impulse is directed to the atrio-ventricular node, or the AVN, because the tissue along the base of the atria is non-conductive, which means that the electrical impulse from the sino-atrial node cannot easily pass directly from the top two chambers of the heart down into the bottom two chambers of the heart. So that wave of electrical impulse has to kind of be funneled through the atrio-ventricular node. Okay, so now we've got the atrio-ventricular node, or the AV node. An important part of this is that there's a short delay between receiving the electrical impulse that spread across the walls of the atria and then sending out a new electrical impulse. That delay is necessary so that the atria can fully contract and that all the blood can be forced from the atria down into the ventricles before the ventricles contract. So it's really important that there's just a slight delay there before that impulse is sent on. The electrical impulse as it leaves the atrio-ventricular node will pass down through the septum, through a tissue called the bundle of His. So this is a bundle of nerve fibers that passes down through the center of the septum. The nerve fibers here are relatively well insulated. That prevents initiation of muscle contraction of the ventricles kind of down the middle first, which is not ideal. So contraction, when it happens through the ventricles, needs to be from the apex up, so from the bottom of the heart up. Once the electrical impulse has travelled down the bundle of His, down to the base of the heart, down to the apex of the heart, is that electrical impulse will start to spread up Purkinje fibers. So this is what's referred to in the specification for OCR as purkyne tissues. It's really the same thing. So purkyne tissues is just a collection of Purkinje fibers. And these are, again, nerve fibers. So they allow the electrical impulse to spread from the apex of the heart up through the walls of the ventricles. And here they are not insulated in the same way as the bundle of His was, and so they will initiate contraction of the ventricles from the base of the heart upwards. This is really important because it will squeeze or push the blood from the base of the heart upwards, causing as much blood as possible to be forced out at as high pressure as possible from the ventricles out into the arteries. We just moved down to slightly different diagrams here. You can see very much the same there, but this one we're just focusing in on showing the movement of the electrical impulse. So from initiation at the sino-atrial node, spreading of the electrical impulse both over into the left atrium as well, but also down into the atrio-ventricular node, not directly down into the ventricles, but down the bundle of His through the septum, and then up branching out into the walls of the ventricles. I know that you've heard this term before where we refer to cardiac muscle as being myogenic or as having myogenic properties. Really what this means is that contraction of the muscle is initiated from within the muscle tissue rather than from a nerve that is coming from outside of the muscle tissue. So you can see that the contraction of the muscle is still being caused by electrical impulses, but that electrical impulse is coming from within the tissue itself. So that's the big difference between this myogenic property of cardiac muscle and the type of muscle that we would see in skeletal muscle.